In this video, we're going to talk about the structure of DNA and the structure of RNA. We may have heard of DNA before, and we know that it's the hereditary material, or what gets passed on from generation to generation. So we're able to see different traits, like you may have the same hair color as your biological mother, your biological father, and that comes from our DNA. Well, DNA is a really, really long molecule, and each segment of DNA that codes for a protein is called a gene. So here in this big long strain of DNA, maybe from this segment to this segment, that codes for protein number 1 to make brown hair. So there's one gene. And then down here we have another gene from this segment to here that maybe codes for eye color. So we have two different segments of DNA that codes for different proteins. Each segment that codes for a protein is a gene. So in our human genome, meaning all of the genes that we have, there's about 30,000 genes. Each cell has every single one of those genes. And humans have about 1 trillion cells in their body. So 1 trillion times 30,000, that's a whole heck of a lot of genes and a whole heck of a lot of DNA. Now let me ask you a couple questions. Do you have lungs in your elbows? No, last time I checked, you only have lungs in your chest. Yet, at the same time, you have the DNA to make lungs, you have the genes to make lungs in your elbows, right? Because we said each cell has all 30,000 genes. So the genes to make lungs you have at your elbows. So my question to you is, if you have the genes to make lungs in your elbows, how come you don't have lungs in your elbows? The simple answer involves gene expression. Gene expression is the activation or inactivation of genes. Basically, are we turning them on or turning them off? If a gene is expressed, the protein that segment of DNA codes for will be made. In other words, the gene is turned on. However, if a gene is not expressed, that means the protein, the segment of DNA that codes for a protein, that protein will not be made, meaning the gene, the segment of DNA that codes for a protein, is turned off. Gene expression is controlled by what we call the central dogma of molecular genetics. And dogma is kind of a big word. Dogma basically means that it's a central law to whatever it is that we're talking about. So here, the central law or dogma, or how things work, of molecular genetics that we're going to get into next. So gene expression is controlled by our one main idea of molecular genetics. And that one main idea of molecular genetics, the central dogma of molecular genetics, is very simple, really. We go from DNA to RNA to protein to traits. So, in other words, our DNA holds all of the information to make proteins. Okay, and going from DNA to RNA, we're going to get more into this later in future videos, but this process is called transcription. Now, the DNA is located in the nucleus, but it's too big to get out of the nucleus. It's kind of like trying to get a semi-truck through the door of a classroom. It's just too big. It's not going to fit through the door. So we have to copy the code from DNA and the information into RNA. That process is called transcription. Now, once we have a copy of the RNA, or of the information in RNA, that information is going to go out to the ribosomes, where the ribosomes are going to make a protein. That process is called translation. translation, going from RNA to protein. And proteins will combine together to make our traits. Proteins come together to make our hair color. Proteins come together to make us tall or shorter. Proteins come together to make our eye color. Proteins come together to see if we have hair on our middle knuckles or not. How many of you just looked? So here's the pieces of the dogma. Remember, we said we started out with DNA. DNA stands for deoxyribonucleic acid. It is a nucleic acid, and it's made of nucleotides, and we'll get into that later, what a nucleotide is. So DNA is too big, it holds all the information, can't leave the nucleus, so we need to make a copy of the information, and we do that in RNA that is small enough to leave the nucleus. RNA is ribonucleic acid. Again, it's still a nucleic acid, and it's also made of nucleotides. The DNA nucleotides and the RNA nucleotides are a little bit different, but they're all still nucleotides, and we'll get into that later. Proteins is what we're trying to make here, and proteins are made of small subunits called amino acids. And then the proteins come together to make our traits. Traits is the characteristics of an organism. And I know that sounds very scientific and kind of lame, but ultimately, different and specific proteins combine for each trait. These are what we see for the most part. So traits are hair color. What's your trait for your hair color? Is it you have a brown-haired trait? Do you have a blonde-haired trait? Maybe you have a red-haired trait? Whatever the case may be. Or your eye color trait, your height trait, you have free earlobes or attached earlobes, that's a trait as well. All of those traits are from the different proteins that were made from the RNA that originated in the DNA in our nucleus. Here you see kind of the general structure of both DNA on the right and RNA on the left. As you see, DNA, we kind of have two of these spiral things. There's one, and here's the other. Okay, we'll get into more of what that's because, but there's two strands in that swirly shape, and then with RNA, there's just one of these swirly strands. So just out of structure alone, you can see that RNA is a smaller molecule than DNA. So let's talk about DNA first. So DNA is the genetic material that contains the blueprints or the information or the instructions for every single protein that can be made in our body and what we need to make. DNA is made up of nucleotides. Nucleotides, okay, so hold on, we're talking about a lot of stuff. We have DNA, which is made out of nucleotides. And those nucleotides break down further into three groups. Those first three groups is a deoxyribose sugar. Okay, so a specific type of sugar that's called deoxyribose. The second thing is a phosphate group. Okay, the second thing is a phosphate. The first thing was a deoxyribose sugar. Second thing is a phosphate. And number three, the third thing that makes up a nucleotide is a nitrogen base. Now, here's another curveball for you. These number three, okay, these nitrogen bases break down into four different kinds. So there's four different types of nitrogen bases. 1, 2, 3, 4. The first type of nitrogen base is adenine. Second is thymine. Third is cytosine, and the last one is guanine. Usually, we simplify these by just calling adenine A, thymine T, cytosine C, and guanine G. This is a nucleotide. Okay, so we have our phosphate, our deoxyribose sugar, and the nitrogen base. The nitrogen base here, you see how there's one ring and two rings. Sometimes nitrogen bases have one ring, sometimes they have two rings. It kind of depends on what specific base that it is. So this base over here will either be an A, T, C, or G. But no matter what, it's going to be the exact same deoxyribose sugar and the exact same phosphate, all to make up this whole thing that is a nucleotide. What is DNA made out of? What does it look like? Well, we know that it has phosphate as part of its nucleotide, and the phosphate is located on the outside of the double helix, or the outside of the ladder. It's that squiggly part I was pointing out before. In between each of the phosphates and each of the nucleotide is the deoxyribose sugar. The deoxyribose sugar is also located on the outside of the double helix, or the outside of the ladder. The deoxyribose sugars and the phosphates will alternate. So it will go sugar, then a phosphate, then a sugar, then a phosphate, then a sugar, and then a phosphate. And you could guess the nitrogen bases are attached to each of the sugars. So the nitrogen bases are on the inside. And again, remember that the sugar, the nitrogen base, and the phosphate all make up a nucleotide. So there's one nucleotide, there's a second nucleotide, and there is a third nucleotide. As we mentioned before, the nitrogen bases that are connected to the deoxyribose sugar are located on the middle of the double helix. If you're thinking of a double helix as a ladder, the nitrogen bases would be the rungs of the ladder. So here's what we're talking about. These are all the sugars and phosphates, sugars and phosphates, sugars and phosphates, with our nitrogen bases in the middle, making up the rungs. Here's another row of sugars and phosphates, sugars and phosphates, sugars and phosphates, with the nitrogen bases being the rungs in the middle. Remember, we said that this is a double helix. Helix is this slinky type shape, and we say it's a double helix because we have two of those strands. These really cool old dudes over here in this really old picture, they are the ones are credited with discovering the shape of DNA and being a double helix. These guys are James Watson and Francis Crick, and they had help of other individuals as well. But we don't need to know James Watson. We need to know that it was Watson and Crick that discovered the structure of DNA. And they did this by looking at a lot of X-ray crystallography pictures and the structures of the DNA. So here we have two DNA strands. And let's just pull these apart a little bit. Here's a sugar, and here's a phosphate, and here's a sugar and a phosphate and a sugar and a phosphate. Notice that we have the sugars are connected to the nitrogen bases. Nitrogen base, nitrogen base, nitrogen base, nitrogen base. All of our C's, T's, G's, and A's. The nitrogen bases are directly connected to the sugar. Over here, we have the exact same thing with our phosphate and sugar connected to our nitrogen base. Phosphate, sugar, nitrogen base. Phosphate, sugar connected to the nitrogen base. And you can kind of see how we have a little bit of a ladder here. So here we'd have our rungs of the ladder. There's our nitrogen base, one on each side, nitrogen bases on the inside. Here, that's connected to the nitrogen bases, we'd have our deoxyribose sugar, nitrogen base, and then in between we would have our phosphates. But the thing about that is it's not a perfect ladder, and more importantly, if this is our ladder, it's a twisted ladder, so twisted into the shape of our double helix. Here's one strand of sugar and phosphates with our nitrogen bases in the middle, And here's the other strand of our sugars and phosphates with our nitrogen bases in the middle. So then we said we had the nitrogen bases that come together in the middle. Well, just to make sure we're all on the same page, if this was a nitrogen base, remember it's part of a nucleotide, so we have our sugar and our phosphate. And it's going to be across, this would make up, the two nitrogen bases together would come in the middle, and they would pair up in the middle. And this is still connected to a sugar and a phosphate to make a nucleotide. Same thing with this, sugar connected to a phosphate to make a nucleotide, and here's sugar connected to a phosphate to make a nucleotide. Now, the thing about it is these guys pair up using what we call hydrogen bonds. They're a special type of bond that holds two different molecules, and in this case, two different nucleotides together. If you see, adenine has 2 spots to hydrogen bond, as does thymine, 2 spots to hydrogen bond. Therefore, no matter what, A's and T's always bond together. So if we know on one strand we have an A, we know that no matter what, there's a T on the opposite side. Conversely, if we look at guanine, we'll see that it has 3 spots to hydrogen bond, 1, 2, and 3, and cytosine has 3 spots to hydrogen bond, 1, 2, and 3. So no matter what, C's and G's are always going to go together. So if we know we have a C on one side, we know that there's a G on the other strand. How everything works is we'll have a phosphate connected to a sugar molecule that's connected to a nitrogen base. And let's say instead of calling it a nitrogen base, let's call it a specific one and call it adenine. And we have another nucleotide in here, so there's a phosphate and a sugar, and let's say we have our G as our nitrogen base, or guanine, and we'll just do 2 more here. So here's one strand of DNA, but remember we said that DNA is a double helix, or it's double stranded. So this guy, this adenine, has to be paired up with another nucleotide of a nitrogen base. So adenine, we said, bonds with thymine. So nothing's changed. It's still connected to a part of a nucleotide with a sugar and a phosphate. We know that this G has to be connected to a C that's also connected to a sugar and a phosphate. And this thymine has to be bonded to an adenine, and it's also connected to a sugar and a phosphate. And last but not least, if we have a cytosine here, we know it has to be bonded to a guanine, or a G, but it's also part of a nucleotide with its deoxyribonucleic sugar and phosphate. So here we have the rungs of the ladder, alternating sugar, sugar, phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar. And here's the other rung of the ladder, going sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, with our nitrogen bases in the middle pairing up together, making up the rungs of the ladder. So here we have our double helix that's twisted, and eventually these things will wrap around and fold around these proteins called histones, and the histones helped everything coil up everything together, and it eventually will form and fold and fold and fold and form this X-shaped molecule that is called a chromosome. A chromosome is nothing more than folded up DNA. So what is RNA then? If we just saw all that stuff for DNA, what's RNA? Well, RNA is still made up of nucleotides, and those nucleotides, RNA, still made of nucleotides, and those nucleotides still break down into three things: a phosphate, a type of ribose sugar, and a nitrogen base. Still same old, same old. Sounds very similar to DNA. The difference is that this sugar that's a part of the nucleotide for RNA is ribose sugar, which is a little bit different than DNA, where DNA had deoxyribose sugar. This is regular old ribose. And again, there's 4 different types of nitrogen bases. So if we take these nitrogen bases, okay, there's 4 kinds. 1, 2, 3, 4. 3 of them should sound very familiar. Okay, we still have adenine, cytosine, and guanine. The difference for RNA is RNA has uracil as one of its nitrogen bases. Remember, DNA had thymine. DNA has thymine, not uracil. Uracil is for RNA only. Lastly, there's 3 different types of RNA that we really need to be concerned with. Well, in actuality, there's really only 2. There's messenger RNA, which we refer to as mRNA. There's transfer RNA that we refer to as tRNA. And then there's rRNA. Right now, we really don't have to worry about rRNA. All we have to know is that rRNA helps to make up parts of the ribosome. Let me write that again. So rRNA makes up part of the ribosome, and that's what the R stands for, is ribosomal RNA. So here's a nucleotide of RNA. Okay, so this is a nucleotide of RNA. We still have our phosphate, our ribose sugar. Okay, if you want to go back, you can look. There's the difference between ribose and deoxyribose. And our nitrogen base. So this whole thing, again, is a nucleotide. And again, the differences between the nitrogen bases could be 1 ring or 2 rings. But these would be our adenine, uracil, cytosine, or guanine, depending on what specific nucleotide that it is. We showed this picture to you before, but ultimately here it is. We have RNA that's a smaller molecule. It's single-stranded, but it's still in the shape of a helix. The sugars and phosphates still make up the outside, with the nitrogen bases making up the inside. DNA is a bigger molecule. It's a double-stranded molecule, or a double helix, so it has 2 strands. Still have the sugars and phosphates around the outside, making up the rungs, with our nitrogen bases, making up the outsides, with our nitrogen bases making up the insides of the double helix. So what's the difference between DNA and RNA? Well, DNA has 2 sides, or it's double-stranded, where RNA is one-sided or single-stranded. DNA has deoxyribose sugar. RNA has ribose sugar. DNA has thymine that bonds with adenine, but with RNA, RNA doesn't have thymine. RNA has uracil that will bond with adenine. Both are still made of nucleotides. Both are still nucleic acids, and they have the same basic structure. And that is all she wrote.
It's like this and like that and like this and it's like that and like this and like that and it's like this. So just chill till the next episode.